Hello, this is Adam Kaeser with the U.S. Fish and Wildlife Service. Thank you for the opportunity to speak virtually to you today about the evolution of low-cost sonar habitat mapping and the exciting prospects for applying this technology to mussel research and conservation in the future in a more time and cost-effective manner. Nearly 20 years ago, the Humminbird Company developed the first recreational grade side-scan sonar system. This low-cost fish finder was capable of producing highly detailed, photo-like imagery of the underwater environment. And though primarily marketed to anglers, other user groups soon took notice. Coincidentally, at this same time, I had landed a job working as a biologist for the Georgia Department of Natural Resources, administering an obscure program focused on the harvest of submerged old-growth timber, otherwise known as deadhead logs. I'd never heard of side-scan sonar before, but one day a deadhead logger invited me out on the water to show off his new side imaging system. His plan was to use it to quickly locate logs in the deep turbid rivers of South Georgia. I convinced my boss to buy one of these devices and began using it to map and quantify deadhead logs and other large woody debris. I wrote an article touting our success that appeared in Fisheries magazine in December 2008. This happened to be the first article describing a research application of low-cost side-scan sonar. Turns out there are many submerged features that can be discerned in sonar imagery. For example, the patch of boulders here surrounded by smaller diameter cobble substrate in the foreground. These substrates appear as coarse and fine textured areas in this sonar image of the exact area. I began wondering whether this imagery could somehow be incorporated into a GIS, where features such as these could be mapped where they occur in real geographic space. Not knowing how to accomplish such a feat, I turned to Tom Litz, who was working at the time as a GIS specialist at Georgia DNR. Tom also happened to be a very skilled computer programmer. Together we began developing a methodology we called low-cost sonar habitat mapping. The basis for sonar image collection was pretty rudimentary in the early days and involved repeatedly capturing screen snapshots while piloting the vessel along a survey course. The side imaging system control head and transducer were small, and the gear could be deployed from small vessels like the one shown here, enabling surveys in relatively shallow waterways. Sonar data could be collected rather quickly, at a rate of 7 to 8 kilometers per hour. Using software tools that Tom created for use in ArcGIS, the sonar screen snapshots were merged and rectified to produce seamless sonar image mosaics. At this point, the imagery could be interpreted, and features could be digitized and classified, producing a habitat map that revealed these features across the landscape. For a period of 5 years or so, Tom and I led workshops focused on training biologists across the country on this low-cost sonar habitat mapping methodology. These workshops were key, as data processing and map development required a significant amount of training and expertise. In the meantime, sonar-based habitat maps were being used to study patterns of habitat use and distribution of aquatic organisms, and a variety of these studies can be found in the literature. Whether side-scan sonar could be used to study freshwater mussels remained an outstanding question at the time I joined the U.S. Fish and Wildlife Service in 2011. A major priority of our field office was to fully describe the distribution and abundance of mussels throughout the largest river in Florida, the Apalachicola. We set out to determine if sonar habitat mapping could help with this major task. We brought Ruben Smit onto this project as a graduate student at Auburn University. The biggest question to tackle was how to delineate habitats throughout a river channel that was almost entirely composed of sand. We began working in the middle reach of the Apalachicola, where the channel meanders in classic fashion. This morphology leads to regular flow crossovers from one bank to the other, leaving what are recognized as flow recirculation zones immediately upstream and downstream of point bars, where deposition of finer substrates occurs, along with increased substrate stability. These recirculation zones serve as flow refugia for fish and mussels, and were the traditionally recognized and targeted habitats for sampling freshwater mussels in the Apalachicola. The recirculation zone can be found adjacent to this bank, highlighted in red. We conducted some pilot scanning at several of these sites, and we're excited to find that these areas did appear different than the surrounding portions of the channel. The red star in this slide indicates where the ladies were standing in the previous slide. Here we found a smooth plain bed form, with darker tones appearing in the upper and lower ends where finer sediments were found. The ability to recognize and delineate these recirculation zone habitats from other areas of the river channel was a major breakthrough. We proceeded to develop a mesohabitat classification scheme based primarily on differences in substrate bed form, recognizing that bed form pattern was indicative of the shear stress forces occurring at the sediment-water interface in sandbed rivers. With increasing shear stress, a flat plain bed form begins to develop ripples and then larger dunes, and these bed form types are also visible in sonar imagery. We hypothesized that mussels might be found throughout the flat plain bed form areas of the river channel based on their association with stable benthic environments, and set out to map these habitats throughout the entire river system. The classification scheme used for the middle reach was informed by concepts developed for alluvial rivers, and sonar imaging was the key to applying and delineating this scheme on the landscape level. The scheme for the middle reach included 5 mesohabitats: the inner and outer recirculation zones, the pool outer bend, the point bar, and mid-channel habitats. Working in other reaches of the system required expanding this scheme to include a few additional classes, for example, habitats formed by man-made structures such as wing dikes. With a map of mesohabitats for the entire river, we developed a stratified random approach to quantitatively sampling mussels throughout the system, a task that unfolded over a period of 4 years as we worked across the upper, middle, and lower Apalachicola and lower Chipola rivers. This graph illustrates the differences in the number of mussels collected in the various mesohabitats, with log mussel density on the y-axis and mesohabitat type on the x-axis. We concluded that 3 mesohabitats, the mid-channel, the point bar, and the man-made structures, were unsuitable for mussels based on the extremely low numbers collected in these habitats. One of the key findings from this study was that we could clearly differentiate suitable from non-suitable mussel habitat by defining boundaries that were apparent when examining bedform patterns. The ability to do so was extremely important to the survey, as 80% of the submerged habitat in this river was determined to be unsuitable for mussels. After solidifying this relationship, we were able to primarily focus the expensive and time-intensive sampling efforts in habitat occupied by freshwater mussels. You can find a lot more of the details and results of this work in the 2 publications identified here. Over the years, a number of important advancements occurred in the realm of sonar data collection and processing. It's no longer necessary to capture screen snapshots. Instead, sonar data can be recorded to a single file through a simple operation. Instead of using ArcGIS to process data, this step can be accomplished using inexpensive softwares. The one I've been using for years is called Sonar TRX. A license costs just several $100s. These new approaches have greatly improved the speed and efficiency associated with producing sonar mosaics. Yet the development of classified habitat maps has remained a manual process. For this primary reason, I believe the field of sonar mapping in inland systems has remained somewhat of a niche field that has not yet realized its full potential in terms of adoption and applications. I began to call the prospect of fully automating the development of habitat maps the holy grail of low-cost sonar habitat mapping. My despair lifted when I had the good fortune to meet and pair Cameron Bodine on the left with Dan Buscombe on the right on a current project focused on mapping potential Gulf sturgeon spawning substrates throughout two large Gulf Coast river systems. Both are high-level computer programmers with a keen interest in side-scan and developing freely available tools. Cam enrolled in a PhD program in computer science at Northern Arizona University, and under Dan's mentorship began working to develop and test automated approaches to sonar habitat mapping. Cam's first step was to refine and complete a piece of software Dan had started to develop. This free program is called PingMapper, and it was written using Python coding language. This program is capable of decoding sonar recordings from any Hummingbird side imaging system. It can export the attributes of every ping on the record and produce sonograms from these data with either the water column present or removed. It then is capable of producing georectified sonar image mosaics. All of these steps are undertaken in automated fashion with minimal user input on the front end. The process of automating the development of substrate maps as a next step is one that uses the AI approach of machine learning to teach the computer to produce maps like a human cartographer. To do so, Cam used a program that Dan created called Doodler to create a training dataset for the computer by doodling over and labeling the various substrate signatures appearing in sonar imagery. The Doodler set was then used to train neural networks in another of Dan's programs called Segmentation Gym. Once the computer was trained to recognize the features it was presented in the training set, it was then programmed to complete the process in an automated workflow. This automated routine has been built into PingMapper version 2. Version 2 of PingMapper is now available for download and allows users to feed in raw sonar data files and output rectified mosaics and classified substrate maps in either raster or vector formats. It's even possible to batch process a collection of sonar recordings in one operation. I believe there is great potential for these automated substrate maps to be used to advance our studies of freshwater mussels. You will note that the scheme developed for the Gulf Sturgeon project includes classes that relate to smooth versus rippled or dune bed forms of fine-grained substrates. Woody debris is also a class that the program will recognize. At the moment, this model has been trained to recognize substrates appearing in the two Gulf Coast systems, but the plan is to test this model in other systems and continue to develop the program to include multiple classification schemes and models that can be selected for by the user. All of this will take time, however, and more resources, and with any luck, Cam and others will continue to work to develop these tools, all of which are open source. On behalf of my co-author, Cam Bodine, I would like to extend our thanks to FMCS for the opportunity to present this work to you, and would also like to recognize some of the agencies that have made this work possible. Our contact information is below. Please reach out if you have any questions or wish to gather some of the literature or supporting materials discussed today. The program PingMapper can be accessed via CAM's GitHub site. Thank you very much.